Hello and welcome to IGCSE Sudarshan Studio. As you have seen the thumbnail, maybe that has attracted you to go for this video, or you're interested in biology, ATP paper, and you have not subscribed yet. That's one of the reason you are watching this video. So if you like the thumbnail, if you like the video, do share and subscribe to the channel IGCSE Sudarshan Studio for the success of IGCSE. Okay, now today we are going to discuss an ATP paper. That's a scanner here. In the end I'll tell you the reason, but this is for the noble cause. Okay, right, for moving to the next one, we are going to solve IGCSE alternative to practical ATP paper, which is for 1 hour. Duration is 1 hour and the marks awarded is for 40, as you can see, 40 marks. Okay, so without further ado, let's go for our paper. 16-page document. Let's go with this paper first. A student investigated the effect of temperature on the movement of water in ciliary stalk. So investigation of effect of temperature in ciliary stalk. The water contained a blue dye. In plant stems such as ciliary stalk, water moves in the xylem tissue within the vascular bundle. So vascular bundle is carrying xylem plus phloem. Both of them together is called VB, vascular bundle. Now the student used this method. First method is, or the first step is, take 2 stalks of ciliary and cut them so that they are 10 centimeter long. 2 stalks of ciliary. Prepare an ice cold water, C and W here. C is for the cold water and W stands for the warm water bath. Place a beaker of a blue dye into each water bath. So the blue dye is of the same level, okay, and in which they kept the large beaker containing cold water. The other one is with the warm water inside the beaker. Fine. Measure the temperature of water bath, water in the water bath, C and W. Let the blue dye equilibrate in the water bath for 3 minutes. So we will be waiting for 3 minutes so that the dye gets equally distributed. Okay. After 3 minutes, place 1 ciliary stalk into each beaker of a blue dye as shown. So in the C and W we have to keep the stalk. After 10 minutes, so here how much duration, that is after 10 minutes, measure the final temperature of water in water bath C and D. So final temperature of the water bath. Okay. Now complete table 1.1 by calculating the change in the water temperature for each water bath. No. Now see, in hurry, what most of the student will do is they calculate the temperature from final. So this is initial and this is final. Directly what you will do, final minus initial, F minus I, that is 15 minus 4. That's true. The answer will be how much? 11. Yeah, the answer will be 11. Okay, it's already degrees Celsius, so no need of mentioning it again, the unit. Now if you apply the same step here, final minus initial, again 28 minus 40, then you get in negative 12, right? But minus 12 can't be in the water change, it won't be in the freezing kind of thing. See, we kept in the water bath, that means the water should be warm. We keep it in the warm temperature, right? So basically it is higher value minus lower value. Rather than final minus initial, we will take higher value minus lower value. So with this is higher value minus lower value, this is correct. Higher minus lower, that is 40 to 28, will give the answer 12. Okay, so C was 11 degrees Celsius and W was 12 degrees Celsius. Fine. Okay, that was the error. Never do in this way, because if it is in this way, why they will give so easy 1 mark? Understand, making you confuse. Next one, remove the ciliary stalk from the blue dye in the water bath C and put it on a white tile. Done. We did this. Cut the small section of the ciliary stalk 5 mm from the end that was in the blue dye, that is from the bottom which was there in the blue dye. From there you cut 5 mm here. End of the ciliary stalk that was in the blue dye, so cut the cross. Fine. Examine the xylem tissue of the cut section of ciliary stalk to see if the blue dye is visible. So you can see the position of the xylem and the blue dye is visible easily here. If the blue dye is still visible in the ciliary stalk, cut another 5 mm. So basically till you see the blue stalk you are going on continuing 5 mm here, and that is what the question is. Continue cutting 5 mm wide section from the ciliary stalk until the blue dye is no longer visible in the cut section. Count the number of sections you have cut from the ciliary stalk that contain the blue dye and repeat the steps 7 to 11 for the W stalk also. Fine. The number of 5 mm width wide section cut for the ciliary stalk C was 4. So the student did this one, and this is the student result. For C stalk, it is ciliary stalk C is 4, whereas ciliary stalk in W is 13. Okay, remember why it is 13. This is W is a warm water. Okay, another one was a cold water, so maybe the warm water is helping in more absorption of water. Is it? We'll see. We'll see that whether the temperature is impacting the absorption of water, or here it is dye. Okay, now prepare a table and record the result shown in figure 1.5. So we have a statement of a student and we need to draw a table, include number of section cut. We have to include in this table section cut, total distance moved by the dye. Okay, so already they have given the title or the heading, so just now we have to draw a table. So first thing first, use proper scale and pencil. So you require pencil and scale, even eraser also by the way. Okay, but don't do with pen. If you use it and if you make some mistake, you cannot erase it. You can't use whitener also. So avoid pen and do with pencil. That would be more easy also. So first thing, we will draw this table. Done. Now we require, one is for the section, number of section, one is for total distance, and one more column we require for stalk in W and ciliary stalk in C, and that one is water bath, right? We kept in water bath, so that means we should keep it here temperature on one side, then temperature is in what? It is in degrees Celsius, right? So here next one is section cut, section number of section cut. Okay, so we'll mention this number of section, number of section. Okay, and the last one that we have is total distance. Okay, so I think I should write, so that's easy. Total distance of what? Total distance of moved by the dye, total distance of dye move per mm. It is in mm. Okay, so we mentioned the unit here. Now temperature, as it is not mentioned in the water bath also, so we will not include. So but what is there in this one? They mentioned cold. okay, or it is C, or you can mention warm or W, both are fine. Number of section, C is having 4 and W is having 13. Number of the total distance of the die move, how much? So how do you give this? Total distance in mm, first one is C. Any idea? Okay, total distance of die that has moved. Okay, there we go. First one will be 20 and the second one will be 65. Can you guess from where we got the 65 and 20? Let's see how much it is. Number of stocks cut here 4. Okay, 5 width, and the other one is 13. Okay, so what we do is, it's very easy now. You just need to multiply that 4 into 5 and 13 into 5. That will give you the total distance. Okay, so 13 into this 5 mm, that the number is, it gives 65, whereas into 5 it will give 5 times 4 is 20. That's it, 20 mm. Okay, from that we got the answer 20 and 65. I hope it is clear now. Be sure you draw it with pencil, use eraser and scale, don't do with pen. And that's our table. Easy. Okay, now moving to the next one. Calculate the rate of movement of the blue dye in their ciliary stalk in beaker W. So rate of movement we have to calculate. What is rate of movement of the blue dye? Rate of movement of a blue dye was 65 mm, total distance moved. What was the duration? Duration for this one was, if you check the last step here, the time duration was around, how much is it? Let's see. Okay, there it is. After 10 minutes, measure the final temperature of water bath of C and W. So it is 10 minutes. So basically what are we going to do is here, 65 divided by 10 will give answer 6.5. But what 6.5 we have to mention? This is in mm and this is in minute. 10 minutes. So the answer is 6.5 mm per minute. Most of the student will make an error here by writing minute as m, and m is a SI unit for meter, not for minute. Okay, so that's an error. Sometimes you students do that. Mm will they by mistake they keep it as m. Okay, I hope this is clear now. The third one. Moving to the next. State a conclusion for this investigation. Conclusion. What is the conclusion of this experiment? See, as the temperature goes high, as this is cold, the distance is only 20. Warm, temperature is more at 10 minutes duration. The same thing that we kept is 65. Number of section 13. Number of section the blue dye is there is 4. That means that higher the temperature, the greater the rate of the dye. So we'll write here, the higher the temperature, the greater the movement of water or what, or dye. Fine, this is what we mention here. That was our conclusion. This investigation was only done once. Explain why it is better to repeat an investigation. Why do we repeat? Why do we do this experiment 2-3 times? And this is to identify something. What is that something? To identify the, let's say, to identify the anomalies. ANO, okay, MALOUS, anomalous results. Okay, to identify the anomalous results, we need to repeat the experiment. But what happens is, no, most of the students during the examination, they consider that giving an average will be the answer for this. Okay, or sometimes they mention that incorrect thought of repeating the experiment will carry out to give an accuracy or a correct answer. Please avoid such kind of things. Okay, repeating of this investigation. Why we repeat the investigation? Not for, I'll use the other one here so that you will get it this point. Okay, we are not repeating the experiment to, and this is error. Okay, most of us will do this one as a common error. And what is that? Experiments are repeated to carry out what? To carry out accurate results. Okay, this is what it is, accurate results or average result kind of thing. This one is completely wrong. We do repetition of experiment to identify the anomalies. Okay, not to carry out or average result to find out. That's completely wrong. I hope that's a clear answer and explanation with this. Okay, so this is the correct one, 1 marks. Next one, identify 2 variables that were kept constant in this investigation. 2 variables. What are basically the variables? We will see in ATP 6 marks question. But variable, please remember, independent is there, dependent is there, and controlled. So 2 variables that were kept constant, something that we keep constant for fair experiment. And what it will be to keep the experiment very much fair? It will be the time. So error here, students just write the word time. They do not complete the statement. What is the time here? Time is basically which time was constant, which time or what purpose they're using. We just mentioned the time there, and that's wrong. Incomplete statement do not award you here the marks. So time, the answer will be time to equilibrate. Okay, so we'll just mention this, time, EQUILI, equilibrate. Or you can mention here length of the time in the dye. Okay, time in the dye. Sometimes student do mention amount here. Okay, never use the word amount. Avoid this thing, amount. Rather than this, replace it with this amount word with volume. Okay, volume of what? Volume of any component, like whichever is given in the example or in the question, but use volume or concentration. Fine. Okay, in this question volume was also there, where volume of water and dye be controlled, but information about this volume of water and dye is already in the question. Okay, volume of the dye, I think they have already mentioned. So volume of the dye and water is already there, so we cannot write the same thing again here because it's already there in the question. Now next one is the data you have recorded in table 1.1 may indicate that there is a source of error. So that's in the table source of error with the method used in this investigation. Identify the possible error and describe improvement to this. What is this error in table 1.1? This is what the table is, right? Okay, this is what our table is 1.1, and what is the possible error into this. So what can be the possible error? Now we have to read this question very carefully. The data you have recorded in 1.1, that means they are relevant to only 1.1 table they are asking. Most of the student will focus on what they will focus on the experimental technique. What experimental technique? Like cutting the stalk or cutting the ciliary stalk for 5 mm section here, position of the die, or not repeating the experiment will one of the error. Like you're not repeating, so I'll just mention here error first. Okay, errors that the student make. First one, like repetition of experiment, or stalk size or length, or position of the die. These are the common things. Okay, and this is not the error part here, because they are relevant. The answer should be relevant to recorded in table 1.1. To that table, the relevant answer, you should focus on the data. Okay, so focus on data here. Okay, so what will be the answer of the error that we have seen? The error will be temperature related. And what is that? Temperature of the water bath not controlled. Okay, so temperature of the water bath not. This is possible error. And how can we improve it? We can improve it by thermostat method. Control the water bath. So thermostatically we can control the water bath here. Okay, that's the error. So repeat the error that is there, this you avoid it, because it's not there. You don't focus on this one, because the question is relevant to the table here, and table related we have temperature there factor. Okay, so temperature of the water bath may not be controlled, so you use thermostat. Okay, now moving to the next bit. In step 9, it is difficult to determine precisely the final position of blue dye. What is the step 9? If you see the step 9 here, examine the xylem tissue of the cut section of a ciliary stalk to see if the blue dye is visible. So the stalk to visit, okay, suggest an improvement to the method that would reduce the source of error. Now student may use the microscope to observe the dye, use the microscope to see the section, but please remember here it is improvement to the method which is given in the question, and it must be relevant only to the question or to the part of the question. So here what it will be related to the section. Okay, how you are dissecting or how you are cutting the stalk. So what it will be, diagonally, vertically kind of thing or something? Do you think that? Okay, so the answer will be yes, correct. The answer will be related to the dissection or cut part, and that will be cut the ciliary, stalk vertically. Okay, cut it vertically so that you can easily find out the content. So okay, that would be the improvement. Or you can cut much more smaller part of the ciliary stalk so that you can get a better result here, but it's for 1 mark, so one statement is enough. Now moving to the next one, D part. Xylem forms cylinder like structure in ciliary stalk. Fine, so cylinder like structure of xylem, it is in this kind of thing. Okay, calculate the volume of cylinder. We have to calculate volume, which is 100 meter long. That means height is given for us, it's 100, has a diameter 0.06. So we need to calculate volume is equals to 3.14, which is already mentioned as a pi value, into r. R is what? Radius. Diameter is this much, so radius should be a half of it, 0.03 into 0.03, okay, into h. H is how much? Height 100 mm, okay, and this is also in mm only. Fine. Now when you multiply all this, so what is the answer that you get is something in the decimal is 0.2865 or something kind of this. Let me check out. Is it the same thing? Yes, 2826, right? Okay, that was there, 2826 in this calculator, 2826. But if we move to the next one here, give your answer to 2 decimal places. So only for 2 decimal place, that means my answer should be 0.28. mm is already given, m and mm, so m cube. Okay, that's how mm into 0.28, 2 decimal places. Our rough work should be clear here. The answer should be mentioned in the given space, 0.28 cube. That's the answer. So we get 2 marks. One is for showing this calculation and one is for correct 2 decimal places. Fine. Okay, clear. Moving to the next one. That's a blank page, and here we go. Celery plants can be damaged by insect pests feeding on them. This can reduce the growth of the celery plants. A black aphid and a larvae of the leaf miner fly are both insect pests that damage the plant. Figure 1.6 is a photograph of a black aphid, which is here aphid, A to B centimeter we have to generally find out. That's how we get the next question. Okay, and the photograph of a leaf miner fly. Now A and B, length of A and B. Now this length of A and B for me is, it is 65. Okay, when I took the question paper printout and all this thing, it was 65, and you can come in the range of the 61 to 65 range. Fine. So this is 65 millimeter or 6.5 cm. Okay, so we'll see what is the next one. It's in mm, so for me it will be 65 mm. I have 6.5 get converted to mm. Calculate the actual body length. That means we have to calculate this part. Magnification is already given, length we already found. What's the magnification? x35. So what are we going to do? 35 equals to 65 divided by actual body length, we keep it as x. Okay, this is our x. Now x moves on this side, left side, 65 divided by 35. What's your answer? Okay, my answer was near to, let me check. Okay, where is it? I did it somewhere. Yeah, it was 1.8. Okay, so when I did it is 1.8, and 1.8, you see this, the next one, give your answer to one significant figure, one significant. So I just round up this 1.8, round off to 2. Magnification, actual body length will be 2 mm. That's easy. 1 marks you get for here, 6.5 get converted into 65. 2 second marks you get for the method of calculation, 1m, and in the end you finally you get 1 marks for writing your one significant figure value. Done. 3 marks distribution. Okay, clear. Moving to the next one. State 3 ways the leaf miner fly shows in figure 1.7 differ from black aphid. So let's spot the difference here. in this two figure? What is the difference? Let's wear the Sherlock Holmes cap and find out the difference. Spot two difference, five difference, anything that you want. Can you? Okay, let's see. The wings are visible. Let's put in this way. What is it? Let's see. The question is, leaf miner fly differs from leaf miner. Okay, though this one leaf miner, we'll put it. Leaf miner has wings. What else we can see? Leaf miner have hair, small hairs. Then they also have antennae, which is very small. I think three things are done. Next one would be, yeah, they have a large compound eye. Okay, large compound eye. Is that fine? Okay, anything else? Legs are short. Okay, and they are also thick also. 3 1 2 3. Okay, anything else you want to see? The overall head, if we see, this is also large. Okay, so we got few differences here, and that difference will mention here. Easy now. If you spot some more difference, you can do that also, but we will see the marks first and then go. See, three differences are enough. So let's write our answer here. Leaf miner has larger head compared to black aphid. Okay, second one, leaf miner have small hairs, okay, small hairs on their body. It is on head region area. Okay, so this is here small head and also on the abdomen, so we'll mention on body. Okay, so small hair on the body, but aphids don't have. But black aphids don't have hairs. Okay, the third and very important one will be, like, let's see, with compound eye we will mention, or wings. Okay, wings are present on leaf miner fly, whereas black aphid don't have wings. Done. This is what we have, 3 points with this comparison. Okay, so we have differed the leaf miner from the black aphid. Whatever is visible, do write that. Hair, head size, okay, antennae, large eye, long hair, short legs, wings are present. The legs are also very thick compared to this one aphid here. Okay, so whichever is visible for you, try to write that, and writing too many points are also not suitable, right? 3 marks for 3 points, that's enough. Okay, moving to the next one. Draw the diagram. Photograph of a celery leaf here. Draw the diagram. Now, first thing, what is required here? Requirement for this one is pencil. Of course, HB pencil 4, which is basically used for diagram purpose, or HB4. Is it HB4? Let me check in the instruction here. I think, see, you use HB pencil for the diagram. Okay, let's go back. So HB pencil, so you can use that, sharp it nicely. Second one is eraser, and third one is a continuous line. Okay, don't lift the pencil, it comes only with practice. So draw the diagram without lifting the pencil. So the line should be continuous and 70% space to be used while doing or while drawing the diagram. Use 70% of space, and yes, no to shade. Don't do shading. No, no, not at all, not at all advisable. Right now, and observe the diagram very clearly. Most of us, we don't observe the diagram here, how the image looks like. Sometimes you make the leaf in this way of your own choice. Okay, though you are drawing the same thing also, it will be on this line or plane like this. So don't do that. As it is, you just need to copy. Don't need to be artist for this. Okay, please remember that we don't require artists to do that. Now only thing, now what we are going to do, the diagram, let me make it short so that even I can do this. Fine. So first thing is the petiole. Check the petiole here. Now I can draw the diagram basically from here also, but I can't see the image then. But still, if I can do, okay, let's see. We'll use the 70% of space this way. Then the leaf goes in this direction. Okay, did I make it? Something we reached up to here. This point is very important here. Okay, why it is important? They see how you are observing the image also. The cleft should be there. Okay, and once we get a cleft here, you can just move your fingers like this and goes in this way. Okay, and then you go in this direction, something in this. Okay, and let's see if this, so online it will be like, let me help you, don't, okay, something in this way. Fine, this is what I get it. So overlapping you should not do, avoid this overlapping to avoid this. Okay, no shading at all. So I'll just erase this part. I hope this, my bad, I have done everything, it goes back. Okay, so fine, and I think no, it is doing everything. So my bad, it's okay, we can't do it much more. Right, so don't overlap like me, that I did here. I will lose mark basically. Now let's see the origin of veins. So one vein which is directly to this part, then this is the first central vein, then we have vein on this way, which is showing to this. Okay, then we have one more vein which goes in this direction and this. Okay, next vein is from this part of petiole, moves on this end, one more is on the next leaf, and let's see how far we can give. Okay, right, these are the veins. Fine, these are the veins of petiole, from the petiole, so all are on the same way. So this is what is there, but I know my examiner, as a part of an this I will lose marks here. One is I think on this area I will lose 1 because I did overlapping. And one more thing I did a mistake is 1, 2 and 3. One more mistake. I will show my mistake, that's better. Okay, maybe you will also do the same thing. So black is better. Okay, so I will lose mark on this side. Of course I have did overlapping, I lose 1. And we have how many? 1 section, 2, 3 larger part. So if I see this is 1, 2 and this is something which is not there in the image also, so I will lose, maybe, I not sure, depends on examiner, but yes, I will lose 2 marks here for 4 mark question. So I will be awarded only 2 and I get -2 because of these kind of things. Okay, so be careful while drawing it. I am showing my error, my mistakes. Maybe you also learn from this. Okay, so 2 marks will be only awarded here. No shading, nothing required. Okay, next, moving to the next question. Learn from the mistake, error. Okay, amylase is an enzyme that catalyzes. Let me first, yeah, I think this is clear. Now, amylase is an enzyme that catalyzes the breakdown of starch to form reducing sugar. Plan an investigation to determine the effect of pH on the activity of amylase. Okay, so now investigation to determine effect of pH is there and activity of amylase we have to see. This keyword why I have underlined, you'll get to know. Now, amylase is an enzyme that catalyzes the breakdown of starch. So, amylase will help in breaking down of the starch to form reducing sugar. That's a statement. We have to plan an investigation and use iodine solution in your method. Okay. Now, before moving to this question, let's see first the background. And what's the background here? We need to check what is independent and dependent variables. Now, before moving to this, let's put first is variable. What are the 3 different kinds of variable? Okay, first thing, variables are of 3 types. What are these 3 variables? Variables are independent variable. Okay, next one is dependent variable. We'll write as VI. No, we will write as independent variable, we'll write as IV. Okay, dependent, we will mention it as DI, dependent variable. And the third one is controlled variable. Okay, CV. So independent variable, controlled variable and dependent variable, right? Now, first thing, what do you mean by variables? This is one thing that we will clarify now. Second one, what to write after the variable. Once we have done with the variable, the next thing comes is method, or we write procedure, how you proceed with the experiment. Okay, now R stands for reliable. Okay, and this is reliability, and S stands for your safety. So whenever you are designing or planning such kind of investigation kind of thing, always remember these 4 letters: variable, method, reliability and safety. That makes VMRS. Virgin males are smart. That's an acronym. To remember, these are the 4 letters which can fetch you for 6 marks. Remember, variable kind of questions are for 6 marks, planning of this. Now, next question is, how do you identify this variable? Which one is dependent and which one is independent? Okay, this also I'll explain here. Let's see. First one is independent variable. What is an independent variable? Independent variable is that variable which you need or you change. Okay, what you change is the independent variable. In this case, what is independent variable? Can you tell me? Is it effect of pH or amylase activity? Yes, it is effect. So most of this, wherever it is written effect of pH, effect of temperature, effect of on the activity or on the amylase activity. So effect of pH, what you change, you are going to change different kind of pH here. Okay, so what you change is basically independent variable. Fine. Like what am I testing for? So I'll just write what I am testing is test. Okay, testing is basically your uh, independent variable, that's pH. Next one is dependent variable. What is dependent variable? Dependent variable is what you measure. In this experiment, what are we going to measure? Imagine, what is the thing that we are going to measure? We are going to measure the time taken the starch to disappear in this activity, right? So in simple way, result I observe. Okay, result that you observe here. Okay, or measure. SULT, result that you observe or measure. And the last one is control variable. Control variable which keeps the test fair. So what you keep the same. Something in the experiment that you keep it constant or same, like for example here temperature, volume or concentration related to this experiment. Okay, temperature, volume or concentration. So I hope this is clear now. How are we going to start our answer? We are going to start our answer by explaining the variable, then descriptions of the procedure, that is method, reliability and safety. These 4 points if we cover, you will get 6 marks. And hope you understand what is the meaning of this independent variable now, dependent variable and constant variable. Once you understand this, what you are going to change is independent, what you measure is dependent, and what are you going to keep safe or same is basically a constant variable. Let's hit the answer now with this point. Let's go. Okay, now first thing is independent variable. Independent variable, we'll mention this part. Independent variable is pH, right? So we'll mention pH, use different buffer. Okay, BUFF here, buffer solution for different pH, that is 4, 6, 7, 8 and 10. Okay, that's what. Second point, so this is done independent. Next one we will do for dependent here. What is dependent variable we are looking for? We are looking for the time for the starch to be completely breakdown. So enzyme activity, right? So that the complete breakdown should be there. So time taken for starch to be completely break down. Then third one, So dependent, independent is done. Next one will be our controlled variable. What's a controlled variable here? We are keeping the temperature constant, that is 37 degrees Celsius. We keep it in the water bath. Then volume and concentration of what? Of starch here, first thing, which is already there. Then amylase enzyme here, and something else, yeah, we are using the buffers for the pH, so we'll mention buffer. No need of remembering the buffer for every acidic, basic and alkaline condition, okay, that we don't have in syllabus. Just write buffer for the particular pH, so that's easy. We have done our part up to here. Volume and concentration of starch. So these are our variable parts are done. Moving to the method or the procedure. So let's go back with our procedure. Okay, so method. What's the method here? First point in the method is place few drops of iodine solution I, okay, iodine solution into wells of a spotted tile. Fine, this is our first point. Second one is add 2 centimeter cube of starch solution to a test tube. Write full thing, okay, don't write just SOL dot, it's solution here. Third one is we need to mention these volumes, okay, always remember to mention the volume. Don't just leave it like add the starch solution into the test tube, add the buffer into the test tube. No, mention the volumes here. Add 1 centimeter cube of buffer solution of known pH. Okay, so whichever pH we are doing with 4, 5, 6, 7, we can do with that. Now next one, place the test tubes. Place the test tube in water bath, okay, in water bath where temperature is, how much is the temperature? Temperature is 37 degrees Celsius, okay. Next one, fifth point, every 20 seconds you can keep it, or 35, whichever timing that you want to maintain it. So basically early if you do the experiment will be fast. So every 20 second, okay, take a drop from the something, no, we missed one point here. And what is that point? It is about amylase. We did not add only amylase and we are doing this part. Without amylase, the enzyme, the reaction won't start, no? So place in the test tube the water bath with the temperature keeping constant for 37, and now add 1 centimeter cube of amylase solution, okay, and mix okay, add 1 ml of amylase solution and mix it and start the stopwatch. Okay, once we start this one, the sixth bit will be every 20 second take a drop of this mixture, the above mixture, in which we have the test tube, we have starch solution, buffer solution, okay, and in that we have amylase also. So every 20 seconds take a drop of mixture and add it to iodine solution, which is in the well, okay. This one. So here it is in the well. Here we already added some drops of iodine in the well in the first step, so do that one. I think this much is clear. And where should I write the further answer? There's no space. Okay, so what I'll do is I'll just erase this part, and you can take a screenshot of this and do it. So no worries. Okay, so if the space is not enough, you can continue on the rough page also, right? Just mention the question number and continue it. So iodine solution placed on it. After 20 seconds we are doing it, and then 7th step is record the time when the we'll mention record the time when iodine solution remain brown. Done. Now we have done with method. M is already done. Now the leftover is reliability and safety. So reliability is reliability is repeat the experiment, okay, and then calculate average result. Repeat the experiment and calculate the average result. You'll get the different time, add them, find out the average. That's what we said. We just mentioned this one. We don't mention the result. We mention reliability, and in the end, so this is R here, and in end we have safety, right? So what is the safety of this experiment? How safely we can do, or safety precaution during this experiment? And the safety is let's see, orally, if I can say, don't mix the dropper with the iodine solution or with the starch or enzyme. Clean the dropper during every step. Or you can mention that use the same volume of starch, amylase and buffer in every trial. Okay, in every trial you use the same volume of starch, amylase and buffer solution. That is one of the safety precaution. Some of the students will write wear goggles, wear gloves, wear coat and all this thing. They are not relevant to the experiment here. Whatever is relevant to the experiment, that safety should be mentioned, and here volume of the starch, amylase and buffer in every trial should be same, or use of a dropper should be different whenever we are following the steps here. So this will give you complete 6 marks, and we have covered VMRS: variables, method, reliability and safety. Remember the virgin male are smart. Okay, that is one of the simple way to remember this part. Or even there is one more thing that is there here. What we have seen, we've seen the starch, buffer, time, amylase, the drop test, iodine there, and the reliability. Okay, I think that's clear here. All your doubts about variable, independent, dependent variables, controlled variables, fine, and how to go with the step. Just follow the VMRS. Okay, next third question here. Scientists investigate the relationship between height and the vital capacity of the lung in people. And the vital capacity is maximum volume of air that can be expelled from the lung after taking the deepest possible breath. Fine. Shows the result of the investigation here. So what is this investigation talking about? Height of a person 130. As the height increases, vital capacity is also increasing. 1.5 to 2.9. Okay, so those first show the result in this. Fine. Plot a line graph, okay, on the grid of the table. Right. So x-axis and y-axis we should first thing is to be mentioned here. Okay, so with this limited space, again things that you should use is pencil, eraser, line graph if it is mentioned line, bar graph it is mentioned bar graph, and units to be mentioned on x and y axis. I suggest my student to write scale also, because for EVM also we need to write the scale. So follow the step of writing scale. No harm if you don't write also, it's fine, but write it properly. Okay, pencil, eraser, and units to be mentioned. So on height is on x-axis, vital capacity, let's put it on the y-axis. So we have to start from 130. That means how many are there? 1 2 3 4 5 6 7 and 8. I can do only 8 here. 1 2 3 4 5 6 7 8. Fine, we start with 130 here. Okay, so this is 130 to, is it like following the series 4 5 6? Okay, 130. Next one is 140, 150, 160, 170, 180, 190, and then 200. This is height. Okay, what is on x-axis? Height of a person in cm. So we'll write here height of a person in centimeter. On the y-axis we have what? We have vital capacity in dm cube, and that is 1.9 and reaches to 7. Okay, 1.9 and reaches to 7. 1 2 3 4 5 6 and 7. So we can use this. 1 2 3 4 5 6 and this is 7. So you can use in this way. Okay, and how is it now? 1.9, 2.2. so let's see 1.9 on this. Okay, this is 1.9, 2.2, something in this way. So just follow this one and then you can do 2.9, 3.4. okay, 2.9, and then next one is 3.4. okay, and what we have 4.1, 5.1, 6.2. 4.1, 4.1, 5.1, 6.2. what else is there? 7.2. okay, so we got 7.2 on this. That means my unit also should be mentioned here, and this is what? Vital capacity in dm cube, right? Done. So we have done with all this part, and now we just need to join the points here. Either you can use a scale or you can use a freehand. Just dark the points there, or you can just put it x over the area. Okay, so best one is to put x only, but if it is not visible, this is also fine. That's it. That's our graph. 4 marks. Okay, and draw the line on this also. Don't forget to draw line on y-axis. I did not do this, so just continue, use a scale, proper scale, and do this part. Okay, don't mess up like me. Next one, describe the relationship here and then use the graph to estimate the vital capacity. Height 175 show on your graph. So on 175, that means this is 170, 175 should be in the center somewhere here, and I have to show it on the graph. So this point related to this one, this is 4 and this is 4.5, that means somewhere it is 4.6. So I'll write here 4.6, 4.7, 1 plus 1 minus is always okay, no problem. So okay, this is what I will show on the graph. 4.7, fine. 4.6 or 4.7 is fine. Okay, here. Next one is describe the relationship shown in your graph between height and vital capacity. So what is happening? As the height increases, the vital capacity also increase. Okay, as the height, we have to give the relationship, right? How the as the height increases, the vital capacity also increases, right? Directly proportional, increase. Okay, now ending with the last question. In people with certain lung disease, the enzyme what is this? Elastase is more active. Okay, elastase is a protein, and fine, in a people certain lung disease, enzyme elastase is more active, and elastase is a protein. State the name of the reagent that is used to test the protein. It's very simple that we need. What is that? Protein. It's a burette reagent. And remember, if it is pyruvate, okay, Benedict solution it is. Okay, solution is for sugar, or you just remember in this way: sugar or diabetes is to person named as Benedict. So Benedict has sugar. Benedict name itself shows that it is a scientist name. So Benedict is suffering from diabetes, that means sugar. Sugar test is for Benedict. Pyruvate is for proteins. Done. So the correct answer is pyruvate here, and we finished 40 marks question. 1 hour, more than 1 hour we took to explain you complete 6 marks question. And by the end of this video, the purpose of the scanner, and this is small short things. In our village there is no school, and our school kids, that is from my village, the school kids walk for 10 kilometers every day during the summer also, just to get the basic education. And my dream, or our dream is to build a school in our village. Every child can receive the education they deserve. Will you help make this dream a reality? Please support our YouTube channel IGCSE Sudarshan Studio as an educational channel, and donate as much as you can. Every child deserves a chance to learn and grow, and together we can bring some difference in their life. Together we can bring smile on their faces. Together we can educate the next future generation. So please support and donate from your heart. Till then, adios guys. Take care of yourself and your parents. Exams are soon, I think in May, June it will be. So do take care of your health, take care of your parents, because they are the most important people in your life. So do scan it, support us, and don't forget, if you are new to the channel, click the bell icon, subscribe button, and share with your friends. IGCSE Sudarshan Studio, a studio where you can guarantee for the success of your IGCSE examination and even for your checkpoint. See you in the next video. Take care. Have a good future.